Hello, everyone. Welcome. So it is my pleasure and honor to introduce Dr. Beatriz Luna as today's speaker for the Perspective in Youth Mental Health series. So Dr. Luna is a Staunton Distinguished Professor of Psychiatry and Pediatrics at the University of Pittsburgh, and she's also professor in the Department of Psychology, BioEngineering, and Radiology, also at the University of Pittsburgh. She obtained her master's and PhD in psychology and did postdoctoral training with John Sweeney at the University of Pittsburgh, and soon after became faculty there. She has many numerous accomplishments, but I will not mention many of them because I want to let her give her talk. But just to mention a few, she's the editor-in-chief of the journal Developmental Cognitive Neuroscience, and she's the founder and acting past president of the Flux Society for Developmental Cognitive Neuroscience, which was launched in 2014. Many of you may know this the Flux Society. And she's the founder and director of the Laboratory for Neurocognitive Development. Her research is about understanding brain mechanisms underlying normative brain and cognitive development across adolescence. It involves using multimodal neuroimaging methods, including REST and task fMRI, DTI, PET, MRS, and many others, and combines them with cognitive testing. She has received numerous and prestigious awards, and the one that I want to emphasize today is the Merit Award given by the National Institute of Mental Health, which is a very prestigious award designed to provide stable, long-term funding to support outstanding, experienced investigators whose productivity is distinctly superior. Her work, with over 200 publications, has had major influences and in many domains, including psychiatry, psychology, neuroscience, and even law, and has influenced and inspired the research of many teams, including mine. And it's really like, I'm so excited that Beatriz is here and to hear your work. And I must say that she's an incredible person as well.
Hi, everyone. Cecilia, you are wonderful, okay? And you've influenced, the way that we think about things. So I'm super excited to be here to let you know about the accumulating work that we've been doing, really trying to understand what are the neurobiological mechanisms that are supporting this very important transition from adolescence to establishing adult trajectories. So we know there's some very unique aspects to adolescence, right? Number one, across societies and across species, there is a peak in sensation seeking. Why is that? A peak in exploration that you'll see really speaks to neurobiological mechanisms that we try to understand. This is also a time when many major psychiatric disorders are starting to appear. Why? When they were younger, maybe there were no evidence that, schizophrenia was in the horizon. And what's interesting is that these two aspects of adolescence share very important things in common, which is that the reward system and the cognitive system are very much involved. So we really focus on trying to understand the neurobiological mechanisms that underlie the maturation of cognition and reward. So overall, the way that we've been looking at development is that initially, from birth to childhood, brain maturation is occurring in a process of accumulation. Your brain is getting bigger, you're getting more cognitive abilities, right? But by the time that you reach adolescence, everything is already there. You even have more than what you'll have later. And then brain maturation is shifting to a process of specialization that will then determine what the basis of your adult trajectory will be and position you for what aging is going to be about. So predominantly in the field, there is consensus that we understand that adolescence is a time of a unique balance. Note that I'm not saying imbalance. There's nothing wrong or broken in the adolescent period, despite what people might think. It is a time when motivational reward systems are predominating over the prefrontal executive systems. And many dual systems models have emerged. Here, the solid line is representing cognitive systems and the hatched line reward systems. And initially, there have been a proposal, oh, maybe limbic structures mature earlier. And maybe that's why, executive systems can predominate. We know that that's not the case. There's continued development of limbic systems. And then there was recognition, hey, there are many aspects of like behavioral phenotypes of, like I said, increases in sensation seeking, but also aspects of dopaminergic function that seem to be peaking during the adolescent period. But still, they were seeing it as a context of, well, but prefrontal cortex and executive function is not there. So what our model is proposing is called the driven dual systems model, is that in fact, adolescence is a time when you have access to adult level executive function. It is not consistent. They're starting to learn how to use it and so forth. It's variable, but you have that access. And then in the context of this increased sensitivity to rewards, one way to think about it is if the brain is saying, all right, you've got everything you're gonna get. Now, let me give you a push with this dopamine. Please go get me experiences that are unique to you so we can make decisions about what your trajectory is going to be as an adult. So what I'll be presenting to you today is evidence that supports this proposal that we have. And we're trying to look under the hood as much as you can in vivo in humans. So please don't expect the elegant work that Cecilia talks about, because I wish we could, but IRB doesn't let us really go in there that much. But we've been looking under the hood, really trying to understand what the mechanisms are. So we want to go beyond just saying, oh, look, more activity in adolescence, less in adulthood. We're trying to understand, wait a minute, what is changing that's underlying this? So, all right, let me tell you what we have found with regards to this curve that we're saying about prefrontal cortex. So we've been doing this for many, many years, and using eye movement tasks. If you guys don't know, Tomas was one of the pioneers of really probing the oculomotor system to understand cognition. In any case, we've been doing this for a long time. We've been looking at inhibitory control, and we say, hey, don't look at this light, right? Look the other way, and it goes like this. Of course, teenagers look at the light, right? To look at inhibitory control. We look at working memory. We say, hey, look at this light. Now come back and remember where that light occurred. And then move your eyes to that location to look at working memory. And we have found a very canonical developmental trajectory of acceleration, kind of like this idea of accumulation. So increases in performance, then deceleration in adolescence. So you're starting to put the brakes on, and then stabilization in adulthood. And we've been finding this for a long time, other labs as well. But we started to think, well, we have these cohorts of 100 subjects, et cetera. Is this relevant, or, are we speaking the same thing as other ways of measuring executive function? And what would big data tell us? So Brendan Turbo-Clemens, who's faculty at Minnesota now, when he was a graduate student in my lab, took this on. And he said, you know what, I mean, he's famous because he's one of the first co-first authors of that Nature BWAS paper saying, hey, you need thousands of imaging data points to look at brain-wide association studies. And what he did here is that he brought in together 4 data sets, 2 cross-sectional and 2 longitudinal. And these data sets were all assessing executive function in their own way. Computerized, different computerized batteries, the eye movement tasks that we have and so forth, trying to see, is there a canonical, developmental trajectory? And what he found that, in fact, that is the case. It doesn't matter how you are measuring it. What we're finding developmentally is this acceleration, deceleration, and stabilization. And in fact, when you apply factor analysis, what is emerging is that there is a general cognitive component that's underlying what you're testing. So what we're really tapping into is something foundational about cognition that has that, type of trajectory. Not only that, we've pushed it because we had more than 10,000 individuals, more than 13,000 longitudinal studies, that we can actually start to say, you know what, by age 18, it seems to be reaching adult levels, which has a lot of implications. Twitter went crazy when we put this out. People excited to actually be able to have something that was a little bit less speculative. Anyway, so that's one aspect phenotypically of how we justify the proposal of how executive function is proceeding. So what's going on in the brain? So as background, this behavioral studies that I showed you before, they're based on actually longitudinal data that is available through the NIH for anyone to analyze and so forth, where we followed people up to 15 years on a yearly basis. Not everyone came to 15 years anyways, but I showed you the behavior results. You can see them there again. And then, but when we looked at brain, what we found, and this is just background, so I'll go quickly, was that the engagement of dorsolateral prefrontal cortex, executive prefrontal cortex, right, was, for correct trials, was used the same way in adolescence as in adult. In childhood more, because it's very difficult, but by adolescence, if you're doing that behavior, you're doing the same thing as adult with regards to PFC. What actually mediated the behavioral improvements was, for example, with inhibitory control, the engagement of, for example, the anterior cingulate cortex. And I see Tomas, and I always tell Tomas, you discovered the anterior cingulate cortex. You was the first one to bring this focus about. And, it is an area that, the way that I like to look at it, it's an alarm. It's like, oh, you did something wrong. Engage cognitive systems, do. And that is continuing to increase through adulthood and explains those behavioral improvements. With working memory, it was the increased engagement of the visual association cortex, because that is where the mnemonic information is stored when doing a spatial working memory task. So the story becomes, okay, prefrontal cortex is there, but it's really a distributed system that really needs to coordinate and engage in such a fashion. But we know, even though we're seeing incorrect trials and so forth, there's specialization that must be occurring in prefrontal cortex. So let me show you a more recent study that we did. This was led by Shane McCune, a bioengineering graduate student who's going to be defending in 2 months. And she was very interested in this working memory task that we did, because there's been a debate. When you're retaining information online, a lot of people, what's really predominantly proposed is that activity is sustained during the delay period to keep that memory. How would that even work mechanistically in a way, right? And there are different theories and so forth. So Earl Miller at MIT, doing monkey neurophys, he found that during this delay period, what's maintaining it are these gamma bursts that are reinstating that memory over and over again until you need to do it in working memory, and until you need to use it. So what Shane did was, okay, we have EEG data. Let's not do the typical, average through trials and see, those sorts of EEG signals. Let's apply spectral analysis at the trial level to see if we can depict these gamma, bursts. And what she found, first of all, and I don't think people can see mine. Okay. The first graph, again, replicating, yes, working memory is getting better all the way into the 20s. And then the first thing that was found was that there was age-related decreases in gamma power. Now I'm emphasizing decreases because I will repeat that again, because we see that as an indication of specialization that I mentioned to you before might be critical in understanding adolescence. Not only that, but she found that there were decreases in the number of the gamma events. So in essence, as you get older, you need less power, less reinstatements of the working memory to end up with better accuracy and faster responses. So that's why I say specialization. You need less. You need optimization of the system, and that's, what must be underlying it. So, well, we know that there are certain things that are occurring in prefrontal cortex that might underlie, these optimization, especially of the gamma frequency, which is very important because it speaks to working memory. So we know, for example, that in prefrontal cortex, you have loss of synapses. And I just remember that we have, all levels of people here. So for those who you don't know, you're born, your neurons make all these connections and they multiply. And then in childhood, it's like, okay, time to get rid of what you're not using and keeping what you're using. So that's synaptic pruning. In the prefrontal cortex, it continues. Actually, lately we've been finding into the 30s. I do like to put a plug for Peter Huttenlocher, who was the one who discovered this, a pediatric neurologist, should have won the Nobel Prize, but he was kind of shy. Because when he came up with this, people are like, what, are you crazy? No, we're not losing neural systems. He's like, well, that's what I'm seeing. So anyways, he's brilliant. His daughter wrote a great book about him. I suggest you read it. It's really great. But anyway, so we know that there are important things that are occurring in prefrontal cortex that have to do with specialization. So in conversations with Bart Larson, when he was a grad student in my lab, now he's also at the incredible University of Minnesota, where they have this, millions and millions of dollars to study development. We used to have these conversations. Hey, people say adolescence is a sensitive period. And I'm like, is it sensitive? Or is it critical period plasticity that we know the mechanisms that will have closing factors that is time dependent? We need to start looking at this. So we wrote a review paper. By that, I mean, he did, right, of course. And, he looked at all the postmortem data, which is predominantly what we know about when it comes to prefrontal cortex and aspects of plasticity, and also animal models. And what he found, oh, no, but first let me, for those of you, critical period plasticity. Here's an example. You're born, you open your eyes, you get all this light in. And that breaks the balance between glutamatergic excitation and GABAergic inhibitory function. When that balance is broken, that is a trigger for the brain to say, oh, shoot, we need to, make some changes and maybe even reorganize that particular cortex so we can recuperate that balance. And, that process is the process of plasticity. And so here's the example of visual cortex. So you open your eyes, you get all this excitation, which increases excitation related to GABA inhibitory function. What's been found is that maturation of inhibitory mechanisms are able to regain that balance. And now that very, very high excitatory information can now be processed. And you have optimal signal to noise ratio. So in what Bart found in this review was that in the postmortem work, there is evidence for decreases through adolescence in aspects of glutamatergic function. And there's also another studies that find that there are aspects of GABAergic function, which has many aspects to it, that are actually showing increases. So, really showing us that, wait, during puberty, these two excitatory and inhibitory mechanisms are actually changing. And it might be reflective of plasticity in prefrontal cortex in adolescence, like really late in development. And we're like, okay, we need to study this. We wrote a grant. We talked to, the leaders in being able to look at GABA and glutamate in vivo in humans. So we had to go to 7 Tesla. So typically neuroimaging is done at 3 Tesla, but we went to 7 Tesla because, as I don't know if you can see a little bit, it's kind of small, but the point is that, the GABA signal is very tiny. Glutamate a little bit better, but also not the best. And, the leading physicists that we were working with said, look, if you really want to look at GABA, You need to control for the macromolecules. You really need to identify that signal to be unique to what you're looking for, GABA and glutamate. So go to 7T and use magnetic spectroscopic imaging, which provides a whole slice with multiple voxels that you can look at, the chemical shifts that are occurring that speak to neurotransmitters. So that's what we did. And Maria Perica, clinical psychology PhD student who is defending next week, doing great work. She took on, writing the paper and so forth. So what we expected was, hey, if there's plasticity, we will see decreases in excitation, reflected in glutamatergic function, and increases in GABA. What did we find? And I love it when our hypotheses don't always go, because I feel like that's discovery. So in blue, I'm showing you for, different parts of the brain. And by the way, the horizontal axis is predominantly age in everything that I present. So you can see in blue that there is a decrease in our measures of glutamate, of excitation. Not a lot of changes in inhibitory control, which we had expected would be increasing. Maybe it's occurring earlier. Maybe we don't have the sensitivity to capture the, compound effect of GABA. But what we really want to know, how is this affecting the balance? It's a balance, imbalance, and then showing balance. And that is in fact what we found. We looked at the correlation between the amount of GABA and glutamate that you had in all these different regions. And we found that that correlation is increasing with age, which is something that, people have used even better than ratio to look at balance. And we're finding that, in fact, oh my gosh, we are seeing in prefrontal cortex this really strong evidence of plasticity that is, starting to end in adulthood. But we took it, to the next level. So here comes Shane. She's like, oh, you guys talk about EI balance all the time. I went to this conference, more neurophys and EEG. And there's a way to look at EI balance looking at EEG data, which all of our subjects who got the GABA glue scanning also had EEG. And this is a way, it's an approach that's called FOOF. Nowadays, it's called SPECORAM, where you look at the exponent of the aperiodic aspect of the EEG signal. So, the EEG signal goes like this. You take that out and you look at this exponent aspect. And what has been found, doing EEG, but also, with postmortem sort of studies and so forth, is that the flatter that it gets represents excitatory inhibitory balance. So she did this, and lo and behold, that's exactly what she found, that with age, we get a flattening of the exponent that is reflective of VI balance. In the next graph, what we find is, let me make sure now I can't even see. Yeah, okay. These same subjects, we saw an association between this EEG measure of VI balance and our MR spectroscopy measures of GABA glue. So really providing even more evidence that what we're seeing are changes in EI balance. And then we see an association as to, the closer you are to the CI balance, the is associated with aspects of your working memory performance as it improves. So then finally, we're really into the CI balance. Maria came over and she's like, you know what? Now there's a new approach with fMRI data doing resting state. So you're inside, you're obtaining brain functional data while you're doing absolutely nothing, where you can apply what's called the Hurst exponent, which is an approach that's been used for many years in Wall Street to predict how stock markets will change. So it's really a way to look at a time series and being able to predict how predictable is this time series as you proceed. So people are starting to use that with fMRI. And in fact, there's at least one study that shows that it's related to EI balance, but in particular to the increases in inhibitory function. So, she's working on this now, but these are the initial results. the Hurst exponent is increasing with age, reflected again of this increase in the eye balance. And it is related to, the variability in initiating a correct working memory response, which improves with age. And I don't show you here, but it is not related to our spectroscopy results, which were in the same individuals. And we've been discussing it, and we think, yes, these are different modalities, but perhaps what we're seeing in EEG and in spectroscopy is really a view of what's occurring in the excitatory systems. And this Hurst exponent might be particularly sensitive to what's occurring with inhibitory systems, which we are not able to capture as much with these other aspects. All right, so one last thing about what might be occurring in prefrontal cortex. We're actually doing something that's structural. And this is led by this amazing postdoc, Valerie Sidner, who came from University of Pennsylvania, from Ted Satterthwaite's lab already with Nature Neuroscience and Neuron Papers, whatever. She's just brilliant. And she's very interested in understanding mechanisms of plasticity. So when you think of plasticity, right, you need closing factors. One of the closing, that says, okay, you're done, you're not going to change anymore. One of them is myelination. So you have myelination, right? You have lipids around the tracts, around the terminals, that not only do they increase, information transmission and provide integrity for it, it's also saying you're done, you're not going to change anymore. So we're very interested in that. And in fact, in the past, us as well as other people have looked at, changes in the tracts of the brain by looking at the integrity of the white matter. And these are very, reliable results. We find that there's, all these changes that are occurring. If you look at frontal systems like the superior longitudinal fasciculus, that big tract seems to be done, by 15 with these measures. But what we did in this old study is that we also went a little bit into the cortex, and we called these regional terminal zones, and we saw those continue to change. So what have we done? So we have this 7T data, which is really exceptional for structure. And we're able to obtain, through transformations from the MP2 rate to the T1 to R1, and I won't bore you with that now. But we're able to get a sort of resolution. First of all, we're able to get a measure of lipids that are very much associated with myelination. And we're able to go through different millimeter depths of the gray matter. So forget the long tracks. Now we're in the gray matter. In trying to understand, are there layer-specific changes that are occurring through the adolescent period? So what, our initial analysis that we've done first is reality check. Are we really able to, are we really measuring myelination? And we're finding that our one results agree with myelin basic protein gene expression levels and with water fraction imaging based myelin. And also we see that the distribution of myelin that you can see here in average myelin with darker colors, more myelin, are distributed the way that we would expect it to be distributed with, more myelin in sensory motor regions. And what I'm showing you all the way to the right are top of the cortex, all the way to deep layers. And we find that, in fact, that's what you would expect. There's more myelination in the deeper layers, right? Because in the top, you just have the ones that are talking to one another, and then, the tracks start coming down, so you have more, and that's exactly what we find. Because of the peel surface and the white matter, we can't get the layer 1, which would be so cool to get, or the bottom of layer 6, but we get other things that could be very much akin to that. So what are we finding? We're finding that there's age-related increases in myelination, and these are greater in the deeper layers. So there's a magnitude effect. There seems to be, more myelination that's occurring in the deeper layers. And let me remind you, the deeper layers are the ones that provide cortical to subcortical connectivity. So top down and bottom up integration of information. And the superficial layers are more cortical to cortical connectivity, providing, localized mechanisms that can support things like working memory, etc., at a different level. So, okay, so we find greater magnitude in deep. And here, I know I'm starting to run out of time, but the other big difference that we find is that in the deeper layers, you see development that's occurring in that shape that I've told you about before. A lot of increases, deceleration, and stabilization. For the deep layers, the superficial layers are showing more linear increases. And we go up to 30 in most of our studies. And some of the thoughts that we've been having are that we always have plasticity at some level in the brain. You get a new car, you have to learn how to use it, or, there's always learning that's occurring, and that's very well understood. And there's beautiful studies looking at myelination during learning, mostly in rodent models, but it's just great. So that's something that exists. And then there's developmental plasticity, which is establishing these trajectories. And perhaps we're able to differentiate these by looking at what's occurring at the layer level. All right, so what we've been finding, looking at GABA glue, EEG, Hurst, and myelination is that, perhaps prefrontal cortex is undergoing this unique plasticity, experience-dependent plasticity that will really play a role in determining what your trajectory is going to be. And if we bring all the other studies together, we think that we're very well justified in our model to propose that that is how prefrontal cortex is operating. All right, so please tell me about time, because I get so into it that, because I want to tell you about the other side of the equation, which is actually, Cecilia, what you'd be more interested in.
Fine.
Excellent. Okay. So now let me tell you about the limbic system. So I already told you prefrontal cortex, right? And then the limbic system, which is predominating over prefrontal cortex. So remember I told you about that task, don't look at the light? And it's such a reliable finding. Many laboratories find adolescents are really not good at it, So what we've been doing now for some years is that we're doing this task, but sometimes we say, you know what, if you do it right, you're going to get more money. And something magical happens. And you can see this in the bar graph. Magically, adolescents are now performing at adult levels. How is that even possible? Something that so many laboratories have found they cannot do. So, and this is just background. What we've been finding now, several different studies using different, reward contingencies, etc. We find that in the presence of reward, and here the graphs are showing you hemodynamic response, those curves. What we find is that adolescents in red, when hey, you might win an award, the ventral striatum goes very, very high. It's like, oh my God, I'm going to get a reward. And at the same time, it pushes the other systems that will allow you to perform the behavior that will give you the reward. In this case, inhibitory control and the frontal eye fields. And we find this in different studies. So it's almost like rewards. Okay, I got to do something really hard. That's all right. We're going to push the system so I can get it done. So we started to think, okay, the striatum, blah, blah, blah. We need to start understanding the reward circuitry a little bit better. So when our next step, which was done by Deepu Murthy at Temple, now he's at Temple now, was to start to look at the functional connectivity between the ventral tegmental area, which is where dopamine is produced, and the striatum. How is that crosstalk occurring? And during resting state, there are no age-related differences. But during a rewarded state, while you're doing this, rewarded antipsychotic, what we have found is that when you're younger and you're a teenager, that connectivity, that coupling is much higher, and it attenuates with age. And we're like, okay, we really need to start to understand what dopamine is all about. So here, back to Bart Larson, who I have to give complete credit. This people are so enthusiastic about this all the time, and it was his idea. It's like, well, we want to look at dopamine in vivo. We don't want to put kids in a PET machine. That's very difficult to do. What do we do? And he's like, well, I've been reading these studies where you look at the iron in the tissue, so not in the blood, in the tissue of the brain. And in fact, he had seen an fMRI study that had really leveraged the fact that there's sensitivity to tissue iron and that they could predict learning. So it might have to do with dopamine. And then a lot of like Parkinson's literature, restless leg syndrome, aging, ADHD, they've been measuring tissue iron to assess the integrity of the basal ganglia. Nobody wanted to say dopamine, however, but we're like, okay, we need to figure this out. So we did the following study. We used a molecular MR machine. So 3T scanner that simultaneously can obtain PET data. So, you know, from 12 to 30-year-olds, we did all the MRI acquisition, including this tissue iron. And then from 18 to 30, we also obtained PET. We obtained a PET measure of presynaptic dopamine with DTBZ, which tags VMAT. So like dopamine availability, how much dopamine you have. And then radiculopride to look at postsynaptic D2, D3 receptor density in the striatum, because we don't know which one is changing, in humans at least. Although that figure on the upper right is a review of what the animal literature shows of how things are changing. And I'll talk to you more about that. So, you know, this is now already kind of like older stuff, but it's such a cool finding. So what do we find? Not surprisingly, tissue iron accumulates with age. That is something we know, especially from aging studies. But in red, you can see that the ventral striatum is starting to show that similar trajectory of increased deceleration and some stabilization. Importantly, when we looked at the association with the more direct measures of dopamine, we found that, in fact, tissue iron is particularly associated with presynaptic measures of dopamine availability. And that makes sense because we know that tissue iron is very important for the production of dopamine as well. So that was huge. And then when we looked at 18 to 30, right, with the PET, we found the following. We found that this tissue iron in the striatum wasn't really changing. It's already been established. But this finding that I think should have way more, you know, people should be talking about it a lot more. We found evidence from 18 to 30 of decreases in the D2, D3 receptor density in the striatum. So what we think might be occurring is the following, is that through development, the brain might be, you know, figuring out how much dopamine are you going to have. And then it says, all right, this is the dopamine that you're going to have. Now, go over there and clean up that mess of all those receptors you have and come up with the perfect match of number of receptors that you need. So kind of akin to synaptic pruning, but at the receptor level. So we've been pursuing this tissue iron a lot. And it can also help us understand what is the balance between prefrontal and the limbic system. So first of all, I'll remind you of the dual systems model proposal that we have. And then Brendan, again, using super big data, he did this for me, for my talks. I love it. In blue is that first study that I showed you, more than 10,000 individuals, how does cognition proceed? And then what he put is a paper that he did independent from me completely, where he looked at a cohort of more than 1 million individuals to look at risk-taking and development. And as you can see, it's looking very similar to what we propose. And I just like looking at it that way. All right. So next, we're like, okay, we have this tissue iron. Let's see where dopamine is contributing. So Ashley Parr, who collaborates with Cecilia and I, and she's really brought us together to do really amazing work. She's like, okay, let me leverage that, what we know about tissue iron. And she was very much interested, okay, decision-making is so unique during adolescence. Let's look at how the nucleus accumbens, right, so the center of reward processing, and its connectivity to decision-making places in prefrontal cortex is changing with development. And let's see if this measure of dopamine function is related to it. And what she found was that with age, there's a predominant, and I like to emphasize, decrease in the coupling between the nucleus accumbens and these different areas of prefrontal cortex. And decrease, again, is the way that we think of specialization, right? We don't know, but we can speculate that perhaps, because, you would think, oh, prefrontal is having more of an effect, it should have stronger connectivity. But perhaps what's occurring is that the influence, the bottom-up influence, and I know it's not direct and connectivity goes through, is maybe being attenuated, like what we've seen with that correlation with VTA and striatum. So, okay, so she found that it's decreasing, but this tissue iron is associated with the strength of the connectivity, even after controlling for age. So that, the more tissue iron you have, which is what you have in adulthood, the weaker the coupling. And in fact, she found that when you look at tissue iron, that mediated the age-related decreases in specialization of frontal striatal connectivity. So going further, she wanted to see how this was related to what we understand of cognition. And what I'm showing you here is that rewarded antisaccade, where, and you can see here in red is doing rewarded trials. You can see that adolescents are performing at adult levels, but in blue, when you don't have a reward, it's that they don't perform as well. And she wanted to understand, well, dopamine should be involved in the fact that there's a differential in response. So what she did, and she's applied this many times now for different approaches, is that she divided the sample into those who had high tissue iron for their age, because it's changing with age, and those who had lower tissue iron. And what she found that it's only those with the high tissue iron for their age who are really taking advantage of this reward aspect to help with decision making. So keep that in mind, because her latest paper that she's working on is using a very big data set that's called NCANDA. It's the precursor of ABCD. And she's looking at substance use. So first she did like very high statistics with, the head of statistics in that cohort and ABCD. And you can see in the first graph, it's what are the patterns of substance use through development. So in red, some people never really use much. I don't know who those people are, but in any case, then there are other ones who take a long time to start to use. But predominantly, there is a peak in substance use in adolescence. That's kind of like normative. When she looks at tissue iron, she finds that those with the less amount of tissue iron for their age are exactly the ones who are experimenting. And I'm going to uncouple this a little bit more, but keep that in mind. Also, she found in a subgroup of these individuals who had done the antisaccade task, again, those with the less dopamine were doing worse in inhibitory control. All right, so keep that in mind. One more study about tissue iron, so we can think about dopamine. So this is Finn Calabro. He's my right-hand person. I can't do anything without him. Seriously, I don't know what I did before he was around. So we did this reinforcement learning task. You see that grid. Each one of those boxes gives you a different probability of rewards. So we had, subjects explore, to see if they could find those that had the greatest reward. And then he applied all these computational models and found, what is the model that best fits the data, including the developmental changes. And what he found was that the best model explains that during adolescence, there is an exploratory, strategy to doing this task. And when you reach adulthood, not exploratory, but optimization of learning. So in other words, they're looking around, they're like, okay, yeah, I know that that one has a lot of rewards, but man, I'm dying to see what that one has. While adults are very much like, okay, I know that's where the big one is, da 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 da, and they're able to move it that way. And this, I think, is really great. And I can't show you all the slides, I've already showed you too much of it. We have other aspects that really speak to adolescence being a time of exploration, behaviorally and brain-wise. Anyway, so then he looked at this factor and how it was associated with the changes in dopamine, in tissue iron. And what he found is tissue iron increases, you see this flip between exploration to optimal decision-making. And again, tissue iron mediates this relationship of changing from exploration to less exploration and more optimal. All right, so this is when I really go off the bend with crazy ideas that I have, trying to make sense of what we're finding. Why is less dopamine associated with more sensation seeking? You would think more dopamine, more sensation seeking, right? How could that be possible? So this is what we've been thinking. So if you think of presynaptic dopamine, how much dopamine do you have? It has that particular trajectory, right, that we've been proposing, but also animal studies have found this as well, that it increases and it stabilizes. And then you have the postsynaptic, how many receptors you have, and that there are animal studies that show that there's a peak, and then, we have these human studies that seem to go with that. So here's the crazy part. So maybe what's occurring is when you're young, you have less dopamine availability. You have all these receptors, and you're supposed to get that system going with not enough receptors and a different match, so a different match, so it can be really stochastic. And perhaps that lends itself to sensation seeking, which may provide a compensatory mechanism. Now, we didn't invent this, or other people like Beta Mogadam and so forth who have said, yeah, in adolescence, that is what's happening. And we're all like, no. But now I'm thinking, oh my God, maybe yes. And then as you get older, or those with more dopamine, and you have fewer receptors, then there's a better match. And you can go ahead and engage that system without, needing extra help to get it going. So, that's just the thought that we've been having. So, okay, in conclusion with this, developmental increases in dopamine function in adolescence influence executive processes. Adolescents with lower dopamine availability have greater connectivity, less inhibitory control, and more exploratory behaviors. And the relative predominance of reward processes over executive function may support the exploration needed for specialization. So again, there's an adaptive nature to exploration that you need in adolescence. All right, so last study is looking at dopamine and how it's related to the CI balance. I'm bringing it back to the prefrontal. And Ashley's been working on this. This is very new. She will be submitting it, she said, by the end of this week, so we'll see. So the way that she's been looking at it, again, she divided high dopamine, low dopamine. And you can see the high dopamine in the darker color. You see age and that change in GABA and GLU. These are all in the same subjects. And what she's finding that it's only those with the high amount of dopamine that are showing this predominant change in EI balance. And it is also those with the higher one that have this particular association with decreases in glutamate. And we know that dopamine, has an effect on excitatory glutamate synapses. So what we think at this point is that changes in GLU-GABA balance in PFC through adolescence may be triggered in part by dopamine's effects on glutamate, so that we can really start to understand, adolescence, you get dopamine might be working, and we had this in the review, might be a trigger. Hey, start specializing in prefrontal cortex, do that balance so that we can really start to integrate new experiences. All right, so now final conclusions. Adolescence is a time of critical periodicity when heightened dopaminergic reward, right? So remember that, heightened, and excitatory processes, also high, predominate over these newly available prefrontal processes, helping in its specialization. And I think one take home that I like to give is that the dynamics of specialization may underlie limitations in cognitive control and heightened sensation seeking, and vulnerabilities to the emergence of psychopathology, but also because it's plasticity, this could be a window of opportunity to influence where that trajectory is going to go. So zooming out even bigger picture, this is how we're thinking about development. You're born with a neurobiological genetic predisposition. It's your ID card, it's who you are. And you have a particular environment. And, through development, especially early on, you're trying to, the brain is trying to make sense of how it's going to be adaptive. And the way that, particularly me, I think of psychopathology, there's nothing broken. It's what you gave it. This predisposition, these environmental demands. And you get to adolescence, and that's a time the brain goes, all right, enough fooling around, enough with the exploration, time to make some decisions. And we have the mechanisms to make that happen. Okay, we have the synaptic pruning, and then we can close this with myelination and perineuronal nets, which I love and I wish I could see, and I'm going to be able to because Cecilia has convinced me that we can do this with the mRNA. She's like, what did I say? So I'm super excited. In any case, and it's really done in a use it or lose it fashion. And the brain just adapts to it. So if you have someone for a deep predisposition for depression in an environment that engages whatever, the amygdala over ventral medial prefrontal cortex over and over again, and the brain is going, oh, okay, we see you use that a lot, we're going to make that predominant, right? Now, if you're really lucky and you have great neurobiological predispositions, a great environment, you end up with a great trajectory. For example, as we know with Cecilia Flores, that's if you're lucky. But when you're not, and that's what we want to understand, these trajectories can differ. Again, I don't see it as broken. I see it as adaptive. And, maybe we can understand a little bit more about pathways to psychiatric disorders that emerge in adolescence. All right, I want to say thank you to my super cool lab and Finn and our funding agencies. And if you liked what I talked about, Cecilia mentioned the Flux Congress. It's in Baltimore this year. It is in Ireland next year. And Brad Schlager, who actually was my vice president of the society, he really helped me make this society. Anyways, he just won the Huttenlocher Award. I won it last year. It was super cool. And he's going to host, and he's the CEO of Kennedy Krieger. So he's going to have lots of resources. It's going to be so much fun. And Margaret Sheridan, who's such an impeccable scientist, has organized the program. All right, guys. Thank you. Questions? Did I use up all my time?
Yeah, thank you very much for a very interesting presentation. I really like the models of having the two curves that intersect at some point. So
yes.
You mentioned something at the end about the psychopathology to understand this. So if you have some developmental-ish psychopathology, schizophrenia, what would your target be? Would you look at the ratio between these two measures, or one developing too early or too late, or one not being strong enough, or with a combination of these?
I think a lot of these things, so some of other work that we've been doing more recently is looking at how this plasticity window can change with time. So for example, with stress, and I know there's a lot of stress models for schizophrenia. And so in schizophrenia, that's, there might be something, very predominant very early on that's emerging during the adolescence. There's the excessive synaptic pruning, theory, which I think could very well be, and that's because one of the reasons is that perineuronal nets have been found to be, impaired in psychosis. So you have this continued, lack of VI balance for a long time that might have effects. So I'm not sure that that answers your question. There's all these dopaminergic sort of things, but it could lend itself to try to understand that.
We talk about the changes in gamma band, but clearly, in adolescence is very high, and adolescence goes lower. Could you explain why and what is it behind?
Well, I think
it is a low gamma or a high gamma.
Great questions, Giovanni. Yeah, so we're looking more at low gamma, although now we have a collaboration with the neurosurgeon and we're doing SEEG, so stereographic EEG, where electrodes are implanted in kids' brains before epilepsy resection, and then we'll start interrogating the high gamma. So we are looking at low gamma. We see these decreases in power, which is what you're speaking about. What could potentially be underlying that? And we're just thinking, low-hanging fruit, synaptic pruning and decreases in glutamate. But maybe you're thinking more mechanistically than that, and I would love to hear it. But the fact is that less is better. I mean, even, the basic aspect of synaptic pruning, you're like, oh, you're losing connection, which is great. Because now, and I don't like using the word efficient because it has these other connotations, but the system is becoming optimized so that you need less. When you had more, it wasn't optimal because it could have gone anywhere, and it was slow and clunky. And then it's a better match. And so you need less power, less gamma bursts.
You have a question over here?
Great work about lots of innovation there, and I don't know of any other place that has developmental data both at 70 and also with 2 events with PET. So I mean, very powerful data sets to work with, possibly tough to get them also. So congrats.
Thank you.
Two points. On the 70 the laminar variations in microstructure, we call it myelination algorithm, but that's still there are variations in cellular composition with myelin certainly playing a role, but there may be other components, and that's why I'm wondering whether you have a chance to look at that separately for males and females.
Yeah, I mean, we just started this process, and I might be wrong, but my memory is that we're not finding sex difference and we're controlling for it. But maybe it's something that we need to look at and look at pubertal timing, which is where the sex differences really start to emerge. But that's really interesting.
Because I would expect differences in the trajectories of those profiles. The other one that caught my attention and it brings me back with dopamine and glutathione. Now, I'm just wondering whether what you see can be interpreted somewhat differently in that from what I remember with glutathione. I remember glutathione. When you have lower binding potential, that reflects internalization of the D2 and D3 receptors. And someone knows that better, just correct me here. But that's how we were interpreting our data that we obtained during brain stimulation. So stimulating prefrontal or motor cortex with PBS, looking at carbon 11 glutathione binding in the striatum. And what we saw that on the stimulation, you see lower binding potential because, in fact, the receptors are internalized when they bind to dopamine. And our interpretation of our finding was that, that's why it's perhaps relevant also to your glutamate, that the stimulation is releasing through corticostriatal pathways, which are glutamatergic, are releasing dopamine locally from the varicosities in the striatum, and that of course drives that internalization of D2D3 receptors. So essentially, glutamate is increasing locally the amount of dopamine, but somewhat paradoxically, it of course then shows up as lower binding potential for replopide. So I wonder, I know that you are not stimulating, but still there is of course a lot of dopamine around under physiological conditions. So could that explain why it's apparently going down, but it's the binding potential that goes down, which may be in fact telling us that there is more dopamine around, that internally endogenous dopamine around that is pushing the D2D3 into the cell, not being available to the fat liver.
I very much love that interpretation that I hadn't thought about. So let me replay it to you and correct me if I'm wrong, and I'm going to make it more superficial. That in many ways you're saying, hey, look, adolescence, due to evidence of maybe increased dopamine function, is in a way we can think about it as brain stimulation, and that what we're seeing with these decreases in raclopride is not fewer receptors, but
it's fewer receptors
available, exactly.
Because
yes, because they were occupied, right?
Yes,
and moved in. Very interesting. Yeah, I need to think about this. Yeah.
That's a very interesting thought, Thomas. And I was thinking similar lines, but in terms of pathology. In schizophrenia, bipolar, one of the well-established findings is high dopamine synthetic capacity in PET studies. And we do see yes, and we do see high sensation seeking among all these populations in terms of substance use, at least cannabis, at least nicotine, there's a higher use compared to the population. And high synthetic capacity, but low active receptor availability, not actual receptor.
So it is that relationship.
Yeah, there is something more to the story.
I mean, we're just putting it out there. And if you have any sort of resting state or task data, you have that tissue iron data. So we did a direct probe in that in that Nature Communications paper using our 2', but we had already written one before in subsequent, where we actually take the T2 star from a resting state or a task, and you average across the trials, and that is very nicely associated with the direct measures of dopamine. And we're writing now a very comprehensive paper on how to do it, because everyone keeps asking us, why do I have that data now? I'm going to look at it, especially if you have clinical populations and so forth. You can go in and interrogate something like that.